Share a little bit about my very my interesting little group. So I actually do my study on gnathiid isopods, specifically the species Gnathia pantherina, and I'll be taking you through their journey from their last meal through to their first breath. And the title might seem a bit strange, but I will explain it shortly. So before we get started with what gnathiids are, I'd like to take you a bit further back to what isopods are. So isopods are small crustaceans, which means they have a hard outer shell, or exoskeleton, which they need to shed in order to grow larger or move on to the next life cycle. Throughout their life, they grow bigger. So as they grow bigger, they shed in each phase. Some species live freely on land, like the woodlice that you might find in your gardens or in aquatic environments like shrimp, while other species might be parasitic, where they are dependent on other host organisms to survive. So the group I study are called gnathiids. These are temporary parasites that live in marine environments. So what makes them especially interesting is that they are not parasites throughout their entire lives, but mainly in their juvenile phases. So these larvae feed on a wide range of organisms, predominantly those from the teleost groups, such as the bony fish and elasmobranchs, which are slightly larger, sharks and rays. When they feed, they attach to their host and drink fluids such as the blood, the tissue fluid, and the blood plasma. And the amount of time they stay attached can vary on fish. It's usually a few hours on the fish, while some of the host attachments on sharks can range up to several weeks. So their appearance also changes depending on whether they're fed or not. As you can see there, the unfed stage, called zoophyia, looks slimmer with more segmented sections on their thorax, or perion, as we like to call it. But as they ingest blood meal, this segment expands and becomes more swollen, making them transition over to the Boknesstrand stage when they are done feeding. So moving on, what does this mean for the host? So gnathiid infestations are not likely to kill their host, but they can cause a number of problems. For example, they may lower the red blood cell count, increase stress hormones like cortisol, or leave wounds that may cause secondary infections. Similar to mosquitoes that transfer malaria between people, they've also been known to transfer blood parasites between different fishes. So smaller fish, like the bony fish, are usually more affected by infestation compared to the larger counterparts like the sharks and the rays. But the sharks and the rays are not completely unaffected. So in stingrays, gnathiid feeding inside the mouth have shown to damage the surface or epithelial tissue, causing inflammation and even lead to excess mucus production and blood congestion. In sharks, they have been documented to damage delicate gill filaments through lamellar disruption. And although the presence of gnathiids did not appear to have a drastic effect on the sharks and the rays, and abundance may cause damage, this damage has also suggested that a prolonged association between the host and parasite is evident. So in contrast to the parasitic larval stages, adults are free-living benthic dwellers. They don't feed. Instead, they focus their energy towards reproduction. And as you can see, the females and the males look drastically different to that of the larvae. And this changes throughout the life cycle. So the life cycle of these gnathiids differ between species and can range from a few weeks to as many as 5 years, which in my opinion is quite long. So a basic representation is as followed. The newly released zoophyia, which are released from the brooding pouch of the female, will look for a suitable host, either teleost or a shark, depending on the species, and then attach and feed for a period of time. After they have fully engorged on a blood meal, they will then release and be categorized as the Boknesstrand stage in search of a suitable host substrate, or in search of suitable substrate like corals and crevices, where they then digest their blood meal and then molt to the next phase, which is common for isopods. So this then makes them transition back to their second stage of unfed zoophyia, which is then goes in search of a new host organism to attach to and feed. And this process generally lasts throughout 3 stages, which they then feed and release and molt until they have reached their third and final fed stage, which is the third Boknesstrand stage, which then transitions to either a male or a female with its final molt. At this point in time, the females and the males reproduce, and the females then release the new round of zoophyia. So my study aimed to elucidate the partial life cycle of the species Gnathia pantherina, with a focus on the third stage Boknesstrand and the adult stages, as well as to investigate their reproductive biology. And this brings us back to the from last meal to first breath idea, as I looked at the gnathiids ranging from their final meal through to the first breath the next generation of zoophyia then took. So in April of this year, 222 third stage fully fed larvae from the species Gnathia pantherina were removed from brown shysharks at Boknesstrand. This species has been documented on the leopard cat shark, as well as the puff adder shyshark, making this the first record for the brown shyshark. The gnathiids were then removed from the buccal cavity, the gills, and the claspers, as you can see in the images, using a brush as not to damage the gnathiids. After removal, they were placed in glass jars, and they received artificial salt water every second day, and were kept in temperature-controlled rooms ranging between 20 and 21 degrees Celsius. Development categories were assigned to the different stages, and the gnathiids were monitored daily for any changes. The development was observed using a stereo microscope and imaging software on either a computer or an iPad, just to facilitate better viewing, as well as to capture images for record. So for the developmental categories, we had 2 main developmental categories between both the male development and the female development. And then the female development was further divided into 2 categories between the juvenile development as well as the female development after she was mature and fertilized. So for the males, we started off with the visibility of the testis, which you can see there in the image. So this typically occurred around 16 days post-detachment, and how they were identified is you can see the testis nicely contrasting against the blood meal. In some cases, it was not as evident because there might have been some change in color, or that we were just surprised with a male. But for the most part, we found an average of 16-day post-attachment leading to testis visibility. As I mentioned previously, molting is a crucial part for isopods to transition to their next phases, and this then brought us to our second developmental phase in the males, which was the posterior molt. So the posterior molt, which occurred about 2 days after the testis, 1 to 2 days after the testis visibility is here. And it's basically like growing a new set of pants, I would associate it with, or new set of legs would probably be the better analogy, which was very interesting to see. This then moved to our third stage, which was the anterior section. So the first developmental stage of the anterior, which occurred around 24 hours after the posterior molt, was visible. We were fortunate enough to capture video footage of an anterior molt, and if you look closely, you can see that what makes these creatures so extraordinary is that they basically form an entirely different head. When compared to the juvenile head, we could see that the males have newly formed mandibles, there where the white arrow is, followed by a change in the cephalosome or head shape, which is basically there with a light blue arrow. As you can see, it enlarged much more. It became more rectangular than that of the juvenile. And if you look at the eyes compared to that of the juvenile's head, the eyes appear much smaller compared to the general head size. Just behind the head, you can see the segmented perion, the second and third pieces, and they also increased in size compared to that found in the juvenile. So the pigmentation after a molt was generally absent. You'll see in a later image that they have these characteristic brown pigmentation on the males, but after a molt, they appeared more clear almost because their exoskeleton did not have the time to harden yet. So they progressively got darker as the exoskeleton got harder with time. So the fourth and final category was passed, or we observed, was that of the red pigmentation visible on the pleonites on the ventral side of the tail. This typically occurred 8 days after the final molt. The pigmentation started light and progressively got darker with time. Furthermore, they were used as an indication of the gnathiids, males reaching sexual maturity, and only after this pigmentation was visible did we place a male with a female. So the female juveniles development was category based on 4 different categories. We ranged them from D1 through to D4, and the D just stands for development. So the first stage was D1 and was assigned based on visibility of the ovaries above the blood meal. On average, it took around 13 days for the host attachment to reach the first signs of the D1. Unlike the testes that do not extend straight through to the posterior end of the perion, the ovaries do extend from the anterior straight through to the posterior. So about 17 days later, the eggs developed to full half of the perion, as you can see there with the second image. And by the D3 phase, which was reached approximately 38 days post-detachment from the host, the eggs had developed to cover the entire blood meal or the whole perion. And it was then at this stage that we placed the females with a male. As you can see here, the male increased in, or the pigmentation visible on the male has become more clear. That's that brown pigmentation that I'm referring to. So because we are unsure at which point fertilization occurs exactly, we deemed it appropriate, or at least in hope of more success, we placed the female with the male at this D3 phase. So after the D3, the D4 was reached, and this was the posterior molt, which you can see in the final image. So basically what happens is half of the perion, or the posterior end of the perion, starts to shed completely, which is quite interesting because unlike lobsters, for example, which start with their anterior end and then move to their posterior end, the gnathiids actually start with their posterior end and then shed their anterior end. You can also see the difference between the adult stage of this D4, which is basically increased in size for the posterior region, while the female still has her juvenile anterior region. So moving on to the next categories, this was predominantly based on the embryo development and started off with the first anterior, or the anterior molt, which then completed the molt overall. As you can see, similarly to the males, the females have a drastic change in their anterior shape. Their whole head is basically also regrown. And yes, our category division ranged from the F1 phase through to the F6 phases. And the F was also just a representation for female. So the anterior molt typically occurred after 24 hours of the posterior molt, which was very similar to the males. This molt is not completed in one session. It is extended over basically 2 days. So when we move on from that, you can see that the eggs for the female first stage still lie on top of the blood meal. And then when they move to their second stage, these eggs then shift to the bottom of the blood meal. So the shift was observed on average of 3 days after the completion of the molt. This was also generally around the time that fertilization occurred, if it did occur. In most cases, we did have success with fertilization. We rarely had an instant where eggs were not fertilized due to placing one male with one female, which we believe increased the success rate. So fertilization of the eggs generally, or fertilized eggs generally took on a round shape, as you can see there in the images below, compared to the eggs that were irregularly shaped prior to being fertilized. At this point, with the F2 stage, females were then separated from the males, and in instances where we had more females that were ready to be placed with males, we then placed them with those males. So when we moved on to the next developmental stages, we started off with the cell division, which became more clearly around 4 days after fertilization. This was then followed by the eye spots visibility, which you can see there, the yellow eye spots that started to be visualized. This also indicates more or less where the head region is, and if you look really nicely, you can almost see the separation of the head from the rest of the body. Yes, this clear pigmentation indicated the next stage that we then placed it in. So for the 5th stage, we looked at the larval tail development. And you can generally see this by the pigmentation that occurs on the tail region. Unlike the head that had the 2 distinct eyes in the front, there was a few more pigmentation spots closer to the tail, which made it easier for me to identify and isolate whether the tails were visible at that point. So at this point, it's also interesting to note that the zoophia already started to indicate pigmentation within the female's brooding pouch, which I think was pretty cool. So as we moved closer or further in the F5 stage, the zoophia began to become more active and started to move. They generally started off slowly. This video that I am showing you was already after they had started being released, and this movement then increases as time progresses, and as the brooding pouch empties, they have more space to move in, which I will show you in the next video. So this video is of a female on the 6th stage who is releasing the zoophia. She released the zoophia posteriorly, and as you can see from the zoophia at the bottom corner, it basically just looks like it's crawling out. So the time, I think, did decrease as the more were released, because there was less competition for the zoophia to be released, if I can put it like that. That stage also typically occurred 60 days after post-detachment. So what we also did was each female was observed to release between 49 and 218 first-stage zoophia. We then also documented a positive correlation between the female's body length and the number of zoophia she released. Obviously, with more space, or not obviously, but with the increase in space for the zoophia, it is possible that it allowed for more zoophia to develop within her brooding pouch. So for the overall study, we were able to document a new host for Gnathia pantherina, which is rather important because we still have a lot of missing information on host utilization. We were also able to establish a bit of a time frame for the different stages. Even though we have around 250 described species, we only have around 7 or so documented life cycles or partial life cycles, which is why it is important to be contributing to more life cycle information, especially to be able to compare between different regions and so on. This is also ideal to base future studies on. So I believe this study in and of itself will be adding valuable insights to future studies as we are working towards a publication. And yes, hopefully this will provide a foundation into future parasitological and ecological studies. So thank you so much for your time. And if you have any questions, please feel free to ask.
Thank you very much, Hesmarié. Really some nice photographs and videos of your parasites. So the floor is now open for questions to Hesmarié. There's currently no typed questions. Is there anyone? So while you're thinking of some nice questions to ask Hesmarié, I will start it off with Hesmarié. You showed in the beginning a photo of the larvae attached to the gill, and then later on you showed some more photos of where they're attached. But there was quite a lot of them on the photo. So my question is, was those photos specifically of a section where there were a lot of them, or is it something that you find regularly that you have actually a very high number of infestation on an individual host?
Sorry. Thank you for the question, Prof. So I think it ranges from host to host. I believe there were some cases where they found a bit less on the host. There have been correlations between the size of the host, though, and the number of infestation or larval attachment. But I do believe it is most likely ranging from case to case. So in our case, obviously, we did have a lot of gnathiids that attached, but I do believe there might be cases with less gnathiids attaching.
Okay. But and in your case, the sharks with, like you mentioned, you had like 200 gnathiids to work with,
Yeah.
which was from 2 sharks or 3 sharks.
Mhm.
Did they, in general appearance, did they look fine? Or any, you did mention some health related issues that has been reported, but do you think those hosts can sustain such a high infestation?
So for the sharks specifically, I do believe they body size allows for higher infestation number compared to if the same number were to be taken off of a bony fish or teleost. I think chances are that they might have a much higher increased reduction in red blood cells compared to that of the shark. So I do think body size is a rather big reason as to why sharks might still be okay after having such a heavy load. Yes.
Okay.
I don't know if that completely answered the question.
No, that's perfectly.
Okay.
Okay. I can't see, people are still trying to digest all the incredible information you
It
gave
is a
through.
lot of information.
I um, can't see any hands yet. Okay, there's a question from Vanessa. She's asking, are hosts being found that have died from such heavy infestation?
So I think mortality is not a very likely occurrence. In cases of fry, for example, it has been documented that as little as one gnathiid have caused the mortality in a fry. But for larger adult fish and sharks, chances of death are not very likely. If it is I think a very, very bad case of gnathiid infestation, I want to call it, that maybe potentially, but it's not very likely.
Okay, great. Then Anya Erasmus asks, what were the main challenges you faced in tracking the life cycle of the gnathids when you did
So
your whole process?
that is a very good question. I think because it was such a new experience and educational experience for me, just there was a lot to learn and a lot I was learning as I was going. Luckily, I did have good literature to refer back to. So I think the largest challenges were trying to catch the molts in time because as I had a general timeline to work with, it still does not pinpoint the exact time a gnathiid might be moving into its molting phase and so on. So some of them did surprise me a bit, but I think once we got the hang of it, it was relatively smooth sailing.
Okay, brilliant. I just think the other thing is worthwhile noting is that you are sitting 6, 7, 800 meters kilometers from the coast.
That's
Um, I,
true.
I did see that in your presentation you mentioned you use artificial seawater. So how did you deal with that, the transition from the seawater that they were originally in, and it seems like your artificial seawater was really good because you mentioned to get them to go through the life cycle, but just maybe some more information on keeping these marine animals alive so far from the coast.
So I think that's when didn't immediately even think of it as being a challenge because our facility has been so useful with assisting with keeping the gnathiids alive. So we do actually have a lovely facility downstairs which has a marine section, and due to this, I think it made it possible to keep them alive so far from the ocean. We did gradually transition them from ocean water that we did bring back, or that was brought back rather, and generally introduced the artificial seawater until they were mainly using seawater. We did have some deformities that occurred within the gnathiids. I believe it might be a natural occurrence because with molds, it might happen where they get stuck. So I don't think that's too much of a surprise. However, the number of deformities might have been due to something that they might have been lacking from their natural environment. So I think if we were to go back to the challenges, that might have then been a challenge actually.
Okay, thank you. I think your answer relates to the next question by Tholoana, who asked, do you think the life cycle takes the same time in natural environment, or could it be faster in the lab because of less competition? Um,
That is a very good question. So just to give a general, or just to move back to firstly the life cycle, we have actually noted that temperature or water temperature might have an influence on the life cycle. So because we had the life cycle in a controlled environment, there was no natural fluctuation of temperatures. So I don't think it'll ever be able to emulate an exact replication of what we find in nature, but I think it's a good start. And then the competition is also a very good point because we obviously decided which males were placed with which females. We reduced the number of competition between the males specifically, so we potentially had higher success rate due to that. But yes, no, I think it would be hard to compare lab conditions to nature specifically, just because you never know what all the factors are going to be within a natural environment. But I do think it's a good baseline to start with.
Okay, great. You mentioned about and the questions also related to competition, and you mentioned that you reduced the competition between males. Did you experience that males actually compete with one another?
So we did do an interesting little experiment where we did place some males together to see if they were indeed aggressive towards each other, and some signs of aggression were viewed. So I do believe that they may indeed compete, and especially because some literature confirms that they have this territorial behavior and form harems of up to 42 females within the harem, I think in the case of Gnathia formica. So yes, no, I think it is possible.
Okay. So Nkosinati says, or has these been observed in specimens in natural science collections? So I think that refers to you mentioned about the deformities that you experience. Nkosinati, am I right? So the question is, do you know if this deformities has been seen in males or specimens from collections from museum material, things like that?
I personally have not seen any collections with deformities. Chances are that in a natural environment, the males that are deformed won't live as long as those that were lived to be up until the point of collection. So I, like I said, personally have not specifically seen any deformities in the posterior region. There may have been some, I wouldn't per se call them deformities, but some differences between anterior regions, but that might just be due to wear and tear with age. So that's, I think, how I see it.
I think I agree with you. I think those ones with the deformities they just don't live enough to be collected long enough. They will quickly disappear from the population, and they're quite popular food. So loads of other fish are known to eat snack on them. So I suspect they will be quickly gobbled up. Another question from Victor. He asked, where do you expect the Pranisa stages to spend their free living time? And then how do they move back onto the shyshark, specifically in Boknesstrand in that environment? Where do you think will they live? And then how do
So
they get on the shark again?
So I believe that these sharks, the general sharks and rays that they target, are those that generally move closer to rocky and coral environments. So basically reef areas, which facilitates in the reattachment of the zoophyte then once it has molt from the Brenisa. Because generally they find a safe space to digest their food and then molt. And they do have the ability to swim a distance, but I think in most cases, these host organisms will generally move close to where they then are. So I think that's what helps facilitate the reattachment.
Okay. Yeah, so safe, you call it safe spaces. It's like in tube worm tubes and within the
Yes.
coral and even in sponges, they can hide away there as well. Okay, so we've got Craig's. Thanks for the great talk. Are you keeping your colony going still? And if so, what is the next step or questions you want to answer?
Thank you. That's actually a very good question, and I think I missed it in the talk. So up until this point, it's not very often that we find any of the first or second stages of these gnathiids that feed on sharks specifically. We generally find the third stage and collect that from the shark. So there's a lot of missing information on what the first and second stages actually feed on. There has been a study that has linked gnathiids that feed on sharks, the third stage, to the second and first stages that feed on fish. But we do not have enough research to facilitate keeping all the different hosts here that could potentially keep a colony going. So unfortunately, that is why we only did a partial life cycle ranging from, like I said, the last meal through to the first breath, because we do not currently know what may be suitable hosts. But apart from that, it's also a challenge keeping a colony alive because the fish tend to eat the gnathiids as well. So you'll have to monitor and facilitate attachment in some way. Not necessarily facilitate, but you need a rather large colony if you would like to keep it going.
Okay, thank you. Yeah, it's always tricky if you don't know what the zoophyte that was just released, what that zoophyte will feed on. I know with the teleost ones, it was easier to complete the life cycle because we know exactly what host they were feeding on, and then they did. And in that cases, there's established colonies. In Lizard Island, there was a colony that lasted for 3, 4, 5 years. And I know in the Caribbean, our colleagues there had a colony that lasted until the last huge big hurricane season went through and blew up everything there. So yeah, so it is possible to keep colonies, but there's no known colony of a shark one exactly for the reasons that you've mentioned. Yes, we've got a hand. Shumelo. Not sure if I pronounce your name correctly. Shumelo Mansi, you've got your hand up. Oh, he's got some mic problems.
You can
Excited.
type the question.
Okay, the hand is down. Let's see. Anyway, I think we had a really nice presentation and a very nice discussion, and thank you everyone for the talks or the questions, and thank you Hesmarié for a great presentation. You've got a few months left for your write-up, so I don't want to any pressure, but we're looking forward to see your final thesis or dissertation in this case. I can also just mention that Hesmarié's first paper from a master was actually accepted with minor corrections, where she described how many new species?
2 new species, Prof, and 1 redescription.
Yeah, so a paper describing 2 new species from South Africa and a 1 new redescription. So yeah, so that is congratulations on that, Hesmarié. Yeah, and then looking forward to seeing some more papers from your work, and I know that Hesmarié is quite keen on continuing with the PhD. So good luck with your future work, and thank you very much, everyone, for attending and being part of this session. Thank you.
Thank you.